Hello, friends. Welcome to my podcast. My name is Dr. Manish Kumar, and today I'll be speaking about metabolomics. I'll be covering topics about what is metabolomics. I'll be also giving you a glimpse on what small molecules are. I will further delve into metabolome and metabolic reactions. I'll also be telling you about importance of metabolomics and how a metabolomics study is designed. So let us start. What is metabolomics? Metabolomics is the large-scale study of small molecules, commonly known as metabolites, within cells, biofluids, tissues, or organisms. Collectively, the small molecules and their interactions within a biological system are known as metabolome. Just as genomics is the study of DNA and genetic information within a cell, and transcriptomics is the study of RNA and differences in mRNA expression, metabolomics is the study of substrates and products of metabolism, which are influenced by both genetic and environmental factors. Metabolomics is a powerful approach because metabolites and their concentrations, unlike other omics measures, directly reflect the underlying biochemical activity and state of cells or tissues. Thus, metabolomics best represents the molecular phenotype. Now, let us try to understand what small molecules are. A small molecule, or also metabolite, is a low molecular weight organic compound typically involved in a biological process as a substrate or a product. Metabolomics usually studies small molecules within a mass range of 50 to 1500 daltons. Some of the examples of small molecules are sugars, lipids, amino acids, fatty acids, phenolic compounds, or alkaloids. There is a great deal of variation in metabolites between species. It is estimated that there are around 200,000 metabolites across the plant kingdom and somewhere between 7,000 and 15,000 within an individual plant species. By contrast, in humans there are thought to be around 3,000 endogenous or common metabolites. These estimates are approximations that are likely to be underestimates because it is difficult to detect low abundance molecules. Nonetheless, it can be concluded that plants are particularly biochemically rich by comparison with many other species. They also typically contain a larger number of genes than other eukaryotes. Well, now let us try to understand what the metabolome and metabolomic reactions are. The metabolome is the complete set of metabolites within a cell, tissue, or biological sample at any given time point. The metabolome is inherently very dynamic. Small molecules are continuously absorbed, synthesized, degraded, and interact with other molecules, both within and between biological systems and with the environment. So, many reactions takes place continuously within cells. So, concentration of metabolites are considered to be very dynamic and may change rapidly from one time point to the next. Current analytical techniques used to investigate metabolomics can only take a snapshot in time under a set of defined conditions. Metabolic reactions. So, metabolic pathways are essentially a series of chemical reactions catalyzed by enzymes, whereas the product of one reaction becomes the substrate of the next reactions. These reactions can be divided into anabolic and catabolic reactions. So, what is the importance of metabolomics? Why is metabolomics so important? Well, the non-invasive nature of metabolomics and its close link to the phenotype make it an ideal tool for the pharmaceutical, preventive healthcare, and agricultural industries, among others. Biomarker discovery and drug safety screens are two examples where metabolomics has already enabled informed decision making. In the future, with the availability of personalized metabolomics, we will potentially be able to track the trends of our own metabolome for personalized drugs and improve treatment strategies. Personalized treatment is likely to be more effective than our current medical population-based approaches. How is metabolomics used? We benefit from metabolomics on various levels, from product and stress testing in food industries, for example, control of pesticides and identification of potentially harmful bacterial strains, to research in agriculture, for example, crop protection and engineering, medical diagnostics in healthcare and future applications in personalized medicine, resulting in personalized treatment strategies. Let us go into bit detail about the applications of metabolomics. As I was saying, in agriculture, biomarker discovery, and personalized medicine. Well, I'll start with agriculture. The development of new pesticide is critical to meet the growing demands of farming. Metabolomics enables us to improve genetically modified plants and help us to estimate associated risks by allowing us to get a glimpse of their complex biochemistry via informative snapshots acquired at different time points during plant development process. Plant metabolomics is particularly interesting because of the range and functions of primary and secondary metabolites in plants. About 300 distinct metabolites could be routinely identified per sample a decade ago, and the number is gradually increasing over time. Uh, Biomarker discovery. So, biomarker discovery is another area where metabolomics informs decision making. Biomarkers are objective indications of medical state absorbed from outside the patient, which can be measured accurately and reproducibly. In metabolomics, biomarkers are small molecules or metabolites that can be used to distinguish two groups of samples, typically a disease and control group. For example, a metabolite reliably presents in disease samples but not in healthy individuals would be classified as a biomarker. Samples of urine, saliva, bile, or seminal fluid contain highly informative metabolites and can be readily analyzed through metabolomics fingerprinting or profiling for the purpose of biomarker discovery. Uh, the third example, personalized medicine. Personalized medicine, the ultimate customization of healthcare, requires metabolomics for quick medical diagnosis to identify diseases. In healthcare, we currently use classical biochemical tests to measure individual metabolite concentration to identify disease states. For example, the blood glucose level in the case of diabetes. Well, metabolomics offers the potential for the rapid identification of hundreds of metabolites, enabling us to identify these disease states much earlier. Now, let us come on to designing a metabolomic study. How do you design a metabolomic study? Well, the two main approaches that can be used in metabolomics are untargeted and targeted approaches. The approach chosen will determine how you design your experiments, prepare your samples, and what analytical techniques do you use. Let us first see what untargeted or global approach is. This method measures as many metabolites as possible from a range of biological samples without any intended bias. This is untargeted or a global approach. The second is a targeted approach. This method is used when you want to measure a set of metabolites and have a specific biochemical question that you want to answer. This approach is often used in pharmacokinetic studies by pharmaceutical industries for drug metabolism and when looking at the effect of therapeutics or genetic modifications of a specific enzyme. Now, let us also understand the complete overview of the workflow of how a metabolomics pipeline or metabolomic studies pipeline look like. So you start with a design experiment, then you go to sample preparation that comprises of direct infusion or chromatography. Then you start with mass spectrometry, then comes the data acquisition, then happens the data processing, and then you finally do the analysis and interpretation. Let us go through each of these main steps in more detail. So I'll be saying a few things about experiment design. The study design, also known as experimental design, is of paramount importance for every study. It is essential to make sure that the samples collected reflect and represent the biology in question. In order to determine and examine the most influential factors that are relevant for the hypothesis under investigation, external factors that can affect the experiment have to be eliminated or identified so that they can be accounted for during data analysis. In the study design, factors like sample size, randomization, and storage must all be taken into account. These help guarantee reproducible and successful experiments that minimize erroneous variability. Noise or error is an important consideration to factor in because it distorts the signals in your data. There are two types of noise: random noise or systematic noise. So, what is a random noise? Random noise results from contaminants and general technological limitations. It produces signal spikes and discontinuous data that could be mistaken for a meaningful data. Systematic noise. Systematic noise results from external factors that are not relevant for the study. Baseline drift is one example of systematic noise and is a common problem in liquid chromatography, mass spectrometry, or LCMS, where the gradient of the mobile phase causes the chromatographic baseline to be irregular. Now, let us understand the sample preparation methods. Sample preparation usually involves the following steps. That is, first you do the collection, then you do the storage, then you do the extraction, then the preparation, or the custom preparation for individual measurement. For example, derivatization for gas chromatography. So extraction techniques. So there are like two kind of extraction techniques primarily. One is solid phase extraction. So solid phase extraction is a process whereby compounds which are dissolved or suspended in a liquid mixture are separated from other compounds according to their chemical and physical properties. Solid phase extraction is often used in metabolomic laboratories to concentrate and purify a sample. Chromatography. Chromatography is an important step in metabolomics experiments to separate individual metabolites from a mixture. The most common technologies used in mass spectrometer are gas and liquid chromatography. Through interactions of analytes with a molecule, through interactions of analytes with a mobile and stationary phase, compounds are separated and elute off the chromatographic column at different time points based on their physiochemical properties. So now let us understand how the analysis is done within the mass spectrometer. So mass spectrometry is an analytical techniques to measure small molecules. The small molecules may be either directly injected into this mass spectrometer or through a coupled chromatographic system. The analytes are ionized at an ion source before they can be detected in a coupled mass detector. The resulting data typically consist of mass to charge, time, and intensity triplets that describe for every detected ion mass the strength of the ion beam and the time it is detected by the spectrometer. So I will not go into details about mass spectrometry, but I'll be telling you also a bit about nuclear magnetic resonance, because this is also one of the techniques that is used to measure organic and some inorganic compounds inside biological samples. So when a sample is exposed to a magnetic field and radio frequency is pulsed, the nuclei absorb and re-emit the electromagnetic radiation. The energy that is emitted as a specific resonating frequency, which depends on several factors including the magnetic properties of the atoms, isotopes, and the strength of the magnetic field. In the case of metabolomics, proton atoms from small molecules are usually investigated. For more, you can of course go much more into detail, but NMR-based metabolomics is a non-invasive and non-destructive techniques with high reproducibility, making it a powerful tool for searching new and novel biomarkers. For NMR, we measure the resulting signal from small molecules, protons resonating with the magnetic fields. One of the first usage of NMR was to detect metabolites in unmodified biological samples. However, when you compare two most common techniques used in data acquisitions, that is the mass spectrometry based metabolomics versus the nuclear magnetic resonance based metabolomics, the difference is very clear. That is, the mass spectrometry based metabolomics is highly sensitive. It's also the number of detectable metabolites is much higher, where you can just, for example, detect 30 to 100 metabolites in NMR, whereas By mass spectrometry, you can detect up to 1,000 or 1,000 plus, depending on whether GCMS or LCMS is used. Now, the NMR is not optimal for targeted analysis, whereas the mass spectrometry based metabolomics is much better for targeted analysis than NMR techniques. When it comes to sample preparation, of course, NMR based mass spectrometry requires minimal sample preparation, whereas for mass spectrometry, you have a more complex sample preparation method. Nuclear magnetic resonance does not require tissue extraction, whereas the mass spectrometry based metabolomics requires tissue extraction. Sample analysis time. Sample analysis time in mass spec based metabolomics is actually much longer because it requires different chromatography techniques depending on the metabolites analyzed. But that is what makes it highly sensitive and reproducible. Well, the instrument cost. The NMR based NMR is actually more expensive and occupies more space than mass spectrometry, whereas the mass spectrometry instruments are a bit cheaper and occupies less space than NMR. Sample per cost, if you see things that is done via NMR, is much low cost per sample, but mass spectrometry is having high cost per sample. But that always gives you an advantage that you can detect much more metabolites. Let us now delve into data processing. So data processing aims to extract biologically relevant information from the acquired data. It includes many steps that are similar for MS and NMR. A good understanding of the steps involved is important in order to minimize this risk of skewed or false results. Typically, the endpoint of MS or NMR metabolomic studies is an feature matrix. A feature is typically a peak or signal that represents a chemical compounds. Thus, a feature matrix contains the intensities, or in other words, relative abundances of relevant signals for every sample, describing the metabolomics fingerprint. Ultimately, this feature list would become a list of identified metabolites with semi-quantified or quantified values. Transpositions of the matrix are also common. To compile a feature matrix, noise reduction and background corrections are essential before features can be extracted via peak picking. These processes greatly tidies up the data. Extracted features of individual samples are then aligned across samples to compensate for the drifts in the chemical shift or retention time in mass spectrometry. So chemical shift is more for NMR and retention time is more for mass spectrometry. Aligned features can then be aggregated in a feature matrix. A feature has a characteristic chemical shift or mass that can be used as a column header. The rows represents individual samples. So to give you a summary of components contributing to signal distortions, a random noise adds variation to the signal around the mean. Systematic noise or baseline shift drifts introduces a systematic drift or a bias in the data that needs to be removed before data analysis. Systematic noise can impact heavily on signal intensities and derived signal areas. The actual signals follows, in theory, a Gaussian distribution. Deviations from the distributions reflect external factors. And finally, overlay of components and the resulting measured signal. So noise is an important consideration to factor in because it distorts the signals in the data. And as I said previously, there are two types of noise. One is the random noise and the other is the systematic noise. So this random noise, for example, results from contaminants and general technological limitations. And it produces signals and spikes and discontinuous data that could be mistaken for meaningful data. Whereas systematic noise are results from external factors that are not relevant for the study. Baseline drift is one of example and is common a problem in liquid chromatography based mass spectrometry or LCMS, where the gradient of the mobile phase causes the chromatographic baseline to be irregular. Now comes the final part, analysis and interpretation. And this is quite complex. So, without venturing too much into the areas of statistic, principal component analysis, and partial least squares, are established methods of, say, multivariate analysis of metabolomics data. So, PCA, or principal common component analysis, is a method that enables us to reduce the dimensionality of the data into inferred variables, thus helping us to identify major trends and features. The dimensionality reduction methods can be used in classification, regression, and prediction exercises. The quality of the statistical models that we infer depends significantly on the data pre-processing, scaling, and normalization methods used. Therefore, successful data requires careful investigation of multiple models for consensus building. In the context of metabolomics, for example, the most common statistical analysis approaches are grouped into unvariate and multivariate methods. Each method offers unique insights into the data structure. Multivariate analysis works on a matrix of variables and highlights characteristics based on the relationship between all variables, whereas the univariate analysis takes only one variable into account, resulting in differently weighted results. The goal, however, of statistical analysis is the categorization and prediction of samples' properties through generation of models that captures the information contained in data matrices. So with that, I would like to end my podcast for today. Hope this was helpful. Thank you. Hope to see you in the next podcast. Bye bye.